Good day, everybody. This is Chris with the Ancient Scholar. I hope this video finds you all doing well. And today what I'd like to do is I'd like to spend a little time talking about the vaccine candidates that are being approved. In fact, one has already been approved, the FDA. That is the BioNTech Fosun Pharma / Pfizer vaccine. Most people just refer to this as the Pfizer vaccine. And then the next one, which is pending approval within the next day or so, is the Moderna vaccine. And these both use a similar kind of technology, and this is somewhat novel technology in that it is the first time that this technology has resulted in a vaccine in humans. The actual technology has been developed or been in development for almost 2 decades. But this is just the first time that it has actually been formally turned into a vaccine. Um, in the case of the BioNTech Fosun Pharma / Pfizer, it is an approved vaccine, and Moderna, as of the making of this video, it's still technically considered a vaccine candidate, but I suspect that it will also be approved. And both of these vaccines / vaccine candidates make use of this same kind of technology with some subtle proprietary differences, and that is these vaccines use what's called messenger RNA technology. That stands for messenger ribonucleic acid. And to understand a little bit about what's going on there, we need to understand something called the central dogma of molecular biology. And I'm going to put quotes around dogma because it's not a literal dogma. When it comes to employment of science, there really isn't such a thing as dogma, quote unquote, in the literal sense, but we say this somewhat figuratively when we say the central dogma of molecular biology. It's just a very, very important idea. And the high yield understanding of this is as follows. You've got a cell, and the cell has a nucleus, and within that nucleus you have the template that cell uses to make all the proteins that it needs. And that template, of course, is contained within chromosomes in the nucleus, and those chromosomes are wrapped up and around and protected in other proteins like histones and so on, but ultimately it contains a code called DNA, deoxyribonucleic acid. Well, nucleic acids in general are very unstable, and they are prone to developing mutations due to exposure to light, exposure to different chemicals outside of the nucleus, different changes in acid base and enzymes. So it's actually pretty fragile. DNA less so than other types of nucleic acids, but still, in human cells, or what we call eukaryotic cells, the DNA stays protected inside of the nucleus under most circumstances. Obviously, if a cell division is occurring, the DNA gets copied, and there are some other things that happen. But under most situations and circumstances, the DNA stays inside of the nucleus. And so what happens is, if the cell needs to make a protein, the DNA gets copied, kind of like taking a textbook and copying a page out of a textbook or something like that. Maybe the analogy that I sometimes use when I teach biology is I use a blueprint analogy. That if you have a blueprint for building say a house, you don't take the original blueprint out to the building site, right? Because if something happens and that blueprint gets damaged, then you're kind of out of luck and you're not able to build the house, right? Because that's the only blueprint you had. So what you do is you make the blueprint and then you keep the blueprint somewhere where it's safe. And instead you make a copy of the blueprint and you take that copy out to the construction site and use the copy of the blueprint. And that is in essence what's happening in the cell. The DNA gets copied. It gets copied into another kind of nucleic acid called messenger mRNA, messenger RNA. And messenger RNA leaves the nucleus, and then it actually kind of hitches a ride on kind of a little highway inside of the cell. And if you look at it under a microscope, it looks like a little lines with tiny little bumps all over them. And this little highway is something referred to as rough endoplasmic reticulum. Reticulum just kind of means highway. And those little bumps that you see are actually little protein manufacturing factories called ribosomes. All right, and so what happens is that messenger RNA takes a ride on that highway until it encounters a ribosome, and then the ribosome reads the code in that messenger RNA, and actually the code, the information coded in the RNA, which is a copy of the DNA in essence, is what the ribosome uses to manufacture proteins. And so this is the central dogma, quote unquote, that is to say that DNA gets copied into messenger RNA, all right, and that is called translation. When DNA gets copied, it gets translated into messenger RNA, or rather, it gets transcribed into messenger RNA, excuse me, and then the messenger RNA gets translated by the ribosomes into proteins, right? So that's a central dogma of molecular biology. Now, various types of viruses can hijack this mechanism. There are some viruses that can hijack this mechanism by injecting DNA. So viral DNA gets injected into the cell, and if that happens, that viral DNA will go into the nucleus, in essence, reprogram that cell into a virus factory, quote unquote. There are other viruses that are RNA viruses, and instead of DNA, they inject RNA into the cell. And typically what they'll also do is they have special proteins that can help convert that RNA into DNA. So they inject RNA and then other proteins. A common protein is something called reverse transcriptase. That's what the HIV virus uses, and that reverse transcriptase, it reverses transcription, essentially turns RNA into DNA. And there are other proteins that act in a similar fashion. And the coronavirus, the SARS coronavirus 2, which is currently what we're dealing with in the worldwide pandemic, is actually one of these RNA viruses here, as opposed to a DNA virus. The SARS-CoV-2 does not use reverse transcriptase, but it uses another kind of protein or an enzyme that can do something somewhat similar. Okay, so that's just kind of the basic understanding. All right, so now that we understand that, and I just want to review very quickly the immune system. And this is going to be relevant to how this particular vaccines work. So when we look at the human or the animal immune system, it is really divided into two major branches. something referred to as the innate immune system. And the innate immune system is the immune system that everybody's born with. And this is just really non-specific. So this just kind of is always there, and it doesn't have any memory. It doesn't have any specificity. It can't identify specific problems. It's just kind of a generalist in the way that it deals with issues. And you have barriers, for example, physiological barriers, mucous membranes, oils on your skin, intact skin, tears, stomach acid, things of that nature that prevent microorganisms from getting into the body. And then you have a group of cells called the phagocytes. And phagocytosis literally means to eat. And these include things like dendritic cells, macrophages, and neutrophils. Some of these are white blood cells, and some of these are very similar to white blood cells. And these cells phagocytize other cells that they detect as being foreign. They can't necessarily detect the exact kind of cell. They can just tell that it's not the human cells. It's something abnormal, and they phagocytize it. And then when they phagocytize it, some of those phagocytic cells, like the dendritic cells, for example, which is kind of an important part of vaccine development, those dendritic cells then can present parts of the microorganism to the other major branch of the immune system called the adaptive immune system, or the specific immune system. And this is the part of the immune system that actually has memory. All right. In addition to physiological barriers and phagocytes, you have what's called inflammation. And inflammation is where you get increased blood flow to an area of the body. You get redness, you get pain, swelling. The capillaries become leaky, so more white blood cells can leak out of them and move into those tissues where there's damage occurring. That's the inflammatory response. And then kind of co-occurring with that, you have something called the complement pathway, which is a bunch of proteins that get activated that can cause various kinds of things to occur, like membrane attack proteins can be formed that can poke holes in microorganisms. You can have opsonization occurring that helps the phagocytic cells better identify those pathogens or those microorganisms. You can get various chemicals that can get released, like interleukins, and that essentially attracts more white blood cells to the site of that injury or that infection. And that's known as chemotaxis, when you have chemical signaling of those cells. Those cells move into the area. Oftentimes they'll attach to the blood vessels, the inner lining of the blood vessel, and they'll just kind of roll along the blood vessel until they slip through a little gap that's opened up, and then they go into the tissue. That's something referred to as diapedesis, or some people refer to that as infiltration as well. Okay, so the bridge between the innate and the adaptive immune system are these antigen-presenting cells. These phagocytes present the antigen, and an antigen is a protein. An antigen is a protein of a microorganism that is specific to that microorganism. It is specific to that pathogen. So an antigen can somewhat be analogous to a fingerprint, right? A fingerprint, right? And that an antigen is a protein that is very specific to a certain pathogen, and that is what the body can use to identify that specific pathogen. That's why antigens, that's why it's so important. It's so critical that these phagocytes can identify proteins that are specific to viruses and bacteria and fungal infections, and then present those proteins to the specific immune system, where ultimately the specific immune system can produce antibodies, and there are some other things that can occur. So when we look at the specific immune system, this primarily consists of what are known as lymphocytes. And there are two major categories of lymphocytes. You've got B lymphocytes and T lymphocytes. We call them B because they mature in the bone marrow. And the B lymphocytes are primarily responsible for something called the humoral response. And this is something that occurs, quote unquote, within the humors of your body. Humors comes from Greek, means body fluids. This is typically extracellular. And this primarily is where we get antibodies that are produced here. An antibody, also known as an immunoglobulin, an IG, that's the same thing, is a special protein that the body produces, and the body produces it in response to the identification of the antigen, right? So the antigen for a microorganism or pathogen is identified, and then an antibody that can bind to that antigen is produced. So antibodies are proteins that are very specific and can bind to antigens, right? And then you've got what are called the T lymphocytes, and these are lymphocytes that mature in response to hormones from the thymus gland. It's near the heart, hence the reason we call these T lymphocytes. And these are responsible for something called the cell-mediated response. And this is important in viral infections in that this occurs within virus-infected cells. And you have two major types. You've got what are called your CD4 positive T cells. That's CD4 is a specific kind of protein. And then you've got CD8 positive T cells. And the CD4 positive T cells are important because they help activate the B lymphocytes, because the B lymphocytes are what produce the antibodies. And so the CD4 help activate the B lymphocytes, and so these are sometimes referred to as helper T cells. And the CD8 T cells, what they do is they actually tell virus-infected cells. They can identify when a cell has been infected by a virus, and then they signal a virus-infected cell to kill itself. They signal cell death. And the cells will die off before they can produce more viruses, right? So when we talk about vaccines, a lot of people really focus on antibodies, which is very important, but it's also critically important that not only do you have a good humoral response with antibodies, but you also want to have a very good cell-mediated response, and that vaccine should hopefully stimulate the CD8 positive T cells, the cytotoxic T cells, to identify virus-infected cells and eliminate them to then prevent the exacerbation of the infection. Okay, so that's a really high-yield overview of the immune system. This is going to be the foundation or scaffolding for understanding how these particular vaccine/vaccine candidates work. All right, so moving on. So these vaccines, what they do, they both have very similar technologies. So what they've done is when we look at the coronavirus, so the basic structure of a coronavirus is you've got a capsule, And then you've got, within that capsule, you've got RNA. All right, you've got RNA inside of that virus. And then you have various proteins on that capsule. And one of the most important proteins is a little protein that kind of looks like this, embedded in that capsule. And interestingly enough, it is this protein that the name of the virus is derived, because corona means crown. And so these proteins embedded in there kind of look like a little crown around that capsule, and that's why these are called coronaviruses. But this protein is very specific to certain kinds of coronaviruses, and this is called the spike protein, or the S protein. And so what we've done is, in January, when the SARS-CoV-2 virus is identified, Chinese scientists then identified and published the entire genome of that virus and the RNA. And it's actually fairly small. It's like 300K. It's a pretty small genome. And then what we did is, quickly after that was published, we found the gene that codes for the spike protein. And then what these companies have done is they have essentially copied those genes, and the gene is the code in the form of RNA. All right. So I'm just going to get rid of this here. And so what they've done is they have copied that, and they've taken that RNA, that really it's messenger RNA that they produced, that codes for spike protein. And spike protein is so specific to the coronavirus that it is a great antigen. Now, you could think that once you could just take that messenger RNA and inject that messenger RNA into the person, right? And then that messenger RNA would go in to the cells and make the cells produce spike protein, and then the body would identify the spike protein as an antigen and start making antibodies and so on. The problem is you've got lots of enzymes floating around in your body, nuclease enzymes, for example, and this is a very uncomfortable environment for RNA. In fact, RNA would not exist very long. If you just took RNA and injected it into somebody's muscle, that RNA would be very rapidly destroyed, in essence. So let me just make that a little smaller so we can see the whole thing. So both of these companies have done is they have actually taken the RNA and they've surrounded the RNA. They've placed it inside of a phospholipid capsule, right? And so that's just a phosphate head with two lipid tails, and that produces a membrane bilayer. And so they have essentially taken this messenger RNA that they've created from the genome, and they've encapsulated it inside of something called a lipid nanoparticle. And that protects the messenger RNA and prevents it from getting destroyed. And furthermore, what happens is when this gets injected into the body, because this is essentially the same structure as the membrane of our body cells, these lipid nanoparticles can then fuse with the lipid bilayer of the membrane of our cells. All right. And when that lipid nanoparticle fuses with the cell membrane, the RNA inside of it will get released into the cytoplasm of the cell. Now, here's the cool thing. This RNA does not go into the nucleus, so it does not reprogram the nucleus of the cell. It does not cause mutations. Okay, so it is actually incredibly safe. In some cases, what we have to do when we make vaccines for certain diseases is we actually have to take like viruses and we modify the viruses to prevent them from causing infection. Or in some cases, we'll take a virus that that maybe only infects certain animals, like chimpanzees, like a chimp adenovirus, for example, and we'll put the genetic material in those viruses, and then that's what we use as a vector for the vaccine. And as you might imagine, that's fairly complex, and if you don't get it right, that can be very problematic. And so in many ways, the messenger RNA approach is very safe, very effective, and does not require a viral vector to work. All right, so you inject the vaccine into the patient's, typically a muscle like the deltoid muscle in the arm, and then those nanoparticles will then fuse with muscle cells, and then they will also fuse with antigen-presenting cells like dendritic cells and macrophages. All right. And so what happens is, in both instances, that RNA is released into the cytoplasm of the cell, and then the RNA ends up going along the rough endoplasmic reticulum where it comes into contact with a ribosome. The ribosome then reads the code within that RNA, and it starts producing spike proteins. Does not produce coronaviruses. It only produces the spike protein. That is the protein that the body can recognize as an antigen. And then those spike proteins end up getting stored in a kind of a storage container called a Golgi apparatus, and then that pinches off, and ultimately those spike proteins, those antigens, make their way back to the surface of the cell, and they will merge with special proteins called major histocompatibility complex proteins, or MHC proteins. And these proteins are what the human body uses to identify self versus non-self. So these are very important proteins, and there are two kinds of major histocompatibility complex, or MHC proteins. There's what we call MHC1, and all nucleated cells, so all cells that have a nucleus, have major histocompatibility complex 1 proteins. So cells like red blood cells, for example, would not have these because red blood cells do not have a nuclei. And then the major histocompatibility complex 2 protein is specific to immune cells, in particular to dendritic cells. And these are antigen-presenting cells that are embedded within your tissues, and macrophage, which are actually monocytes, which are a type of white blood cell. These are monocytes that leave the blood and go into the tissues, and when monocytes leave the blood and go into the tissues, they are referred to as macrophage. And so the dendritic cells and the macrophage have MHC2, whereas all nucleated cells, including those muscle cells where these lipid nanoparticles merged with, have major histocompatibility complex 1 proteins. Okay, so moving along. So let's talk about major histocompatibility complex 2, MHC2. So the spike protein, after it is produced, attaches to the MHC2 receptor, and then you have, let me make that a little smaller so it's a little easier to see. You have a T cell, specifically a CD4 positive T cell comes in, and it actually interacts with that spike protein here through something called a TCR protein or a T cell receptor protein. And then the CD4 protein also helps the white blood cell attach to the receptor. And essentially that spike protein, the CD4 positive cell, recognizes that, and it then kind of becomes an activated. It starts off as what we call a naive cell, and then it becomes an activated cell, and it becomes an active helper T. And when it is activated by that spike protein, by that antigen, it begins releasing lots of chemokines, various what are called interleukins. Inter means between leukin, white blood cell, like interleukin 2, interleukin 4, interleukin 5, etc. And these chemicals then act upon the what we call the B lymphocytes. So this is the T, the helper T, then acts upon the B lymphocyte and causes the B lymphocytes to proliferate. So you get lots of them, and then they differentiate, right? They turn into their active form, which is called a plasma cell, and they begin producing lots of antibodies. And then what those antibodies can do is, if you have a coronavirus here with spike proteins, those antibodies bind to the coronavirus via attaching to those spike proteins, because again, these antibodies come from activation by the helper Ts, which have ultimately been activated by the spike protein used as an antigen, right? So the antibodies are, those specific antibodies are very specific to the spike protein of the coronavirus, right? And then those antibodies can attach and they can deactivate the coronavirus, they can opsonize it, surround it, allow other immune cells to come in, they can cause the virus to fall out of solution. There are lots of different things that antibodies do. That happens is through major histocompatibility complex 1 or MHC1, you have your other type of cell, all right, your CD8 positive T cell, which will ultimately become a cytotoxic T cell. And so it attaches to MHC2. And again, this is the spike protein, which is acting as an antigen. So you've got your TCR protein that binds here, and then you've got the CD8 protein, which helps stabilize and bind to the major histocompatibility complex 1. And then the naive cell is activated when that occurs, and it turns into a mature cytotoxic T cell. And what it can do is it can also release lots of chemokines, lots of signaling molecules. And in addition to that, it will signal cell death of virus-infected cells and prevent virus-infected cells from producing viruses. So at the end of the day, you have a battle going on two fronts. You've got the humoral part of it, which are the antibodies, and then you've got this cell-mediated part, which are the identification and destruction of virus-infected cells. Okay, And so that's what these that's how these particular vaccines work. I've had a chance to look at the data from the Pfizer vaccine. I have not looked at all the data on the Moderna vaccine. Just it's a lot to go through. It's about 100 pages. I think it was 98 pages in total on the Pfizer vaccine, and it's out there. You can actually download it. It's open access to the public, so you're more than welcome to find that and look at it. But it is incredibly compelling. Uh, the trial consisted of almost 40,000 people. It was a true randomized controlled trial, or an RCT. So you had a control group that received essentially an intramuscular injection of normal saline, and then you had the intervention group that received the vaccine. And the COVID infection rates were similar in both groups till about 10 days after vaccination, at which there was a substantial parting. In fact, the intervention group that received the vaccine essentially flattened completely out, and after about 10 days, there were virtually no infections, whereas the placebo group spiked up substantially, and so we're sitting at around 95% efficacy for the Pfizer vaccine. So it's pretty remarkable. Um, the side effects that we've seen so far are fairly typical side effects for viruses, and typically when it comes to vaccines, virtually all of the substantial side effects that we ever see with vaccines occur within the first 6 weeks of someone receiving a vaccine. Um, and we are now a few months into this, so we're well over that 6 week mark. And so the safety profile appears to be fairly well understood. Obviously, there's an argument for long-term issues um, that that can be appreciated and talked about. But with that in mind, it does appear to have a pretty favorable safety profile. Of course, allergic reactions are always possible anytime you put a foreign material into somebody's body, and we have seen a few of those. And there have been some cases of Bell's palsy reported, but the thing about that is I don't know that you've got to be able to tease that out of the background rate of Bell's palsy. Bell's palsy is not a rare problem, and there's a certain amount of people that are just going to get Bell's palsy, typically from viruses like certain herpes viruses can cause Bell's palsy. It's a fairly common problem. So it's not clear to me that there is any relationship between the vaccine and Bell's palsy, but it's certainly something to look out for. So we'll see what happens. But all of the indications currently are that this is a very effective vaccine with a pretty good safety profile at this point. So hopefully you all found that interesting, and I'll try to get this video posted. I'm actually so where I live, we've had an incredible spike in cases over the past couple of months. The situation's actually pretty terrible, if I'm being quite honest. And they ended up shutting the college down. In fact, we are no longer even able to go into the hospitals or labs or anything like that. And so I'm actually doing COVID contract work. I'm taking care of patients in essentially in makeshift hospitals because our hospital capacity statewide is essentially full. We essentially don't have space for COVID patients throughout the state. So we've been opening up makeshift hospitals to help care for the increased load of patients. And so I'm working at one of these doing night shifts. Um, so I actually had some time to do a video because I'm not teaching currently. So I thought I would do a video on the vaccine. And again, all apologies if I sound tired. It's been a while since I've worked night shifts. Um, so yeah, I used to work nights quite a bit back when I was a newer nurse in my 20s. Um, but now that I'm in my 40s, I'm finding working night shifts to be a little more challenging as far as the sleep-wake cycle goes. So I'm having to try to adjust my circadian rhythm, and it is a little challenging to say the least. So I am just a little bit more tired than normal. But anyway, I hope this video is still helpful and informative, and I hope you all stay safe and healthy going into the holiday season. Take care, everyone.